All right, hi everyone. We're back to finish part 2 of our biochemistry lecture. This part's going to be a lot shorter. I think the last one was like 30 minutes. We just have a few to get left, a few left to take care of. So, starting off with lipids. Lipids are fats, right? They're made up of a bunch of fats. So, background information: they are non-polar organic compounds. So, they're organic, meaning they have a bunch of CHCO, and they are non-polar, meaning they're not going to have that charge that we see on polarized molecules. Also means they're not going to dissolve in polar solvents, specifically water that we find in the body, because they're not making those hydrogen bonds to that polar solvent. They don't have that charge. Like carbohydrates, they are composed of CH and O. With lipids, we see a greater CH ratio. So if I go back here to, let's pull up this sugar down here, this glucose. I think I need to turn my pen on. Specifically, this is glucose, so one of our simple sugars. We have 1, 2, well, if we're counting these, we'll start 1, 2, 3, 4, 5. So this would be C1, C2, C3, etc. You see on this first carbon, it is bound to one hydrogen. It's also bound to this. It's called a hydroxide. Anything OH is a hydroxide. If it was on its own, it would be a hydroxyl. Here. Okay, so we have another hydroxide, another hydroxide, another hydroxide. Here, carbon bound to another carbon, which is bound to two hydrogens, and another hydroxide on the end. So we have, and then finally this last bond, carbon to oxygen, oxygen to carbon. When we say lipids have a greater carbon-hydrogen ratio, that means they're going to have a greater energy density. So when you're looking at these structures, each one of these corners is going to be a carbon, 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 all the way down. And because it has a bond to a carbon here, a bond to a carbon here, it's also going to have two hydrogens on each of these carbons. We don't need to draw that in this structure because it's inferred. It's something you just have to know when you take chemistry or when you take another chemistry class. So each one of these carbons, except for down here, is bound to two hydrogens. This last one, it has one bond with another carbon, so it's going to have three hydrogens here. If we were looking, this is a saturated fatty acid. That means pretty much every carbon is going to be bound to two hydrogens or three hydrogens at the end. Here, if we're looking at monosaturated, what saturation is talking about is this double bond right here. So here, we have carbon sharing two electrons with this other carbon, and then we're only going to have one hydrogen off of each of these carbons. So saturated, monosaturated, polyunsaturated, we're going to have more than one double bond. There's three types of fat, lipids: neutral fats, phospholipids, steroids. You've probably heard of at least two of those. Maybe you haven't heard of neutral fats. You've probably heard of the term triglycerides. So triglycerides are how we store a lot of energy in our body. They provide insulation through subcutaneous fat, energy storage. You see there's the most concentrated source of usable energy in the body. That's a good test question. Cushioning for internal organs, absorption of fat-soluble vitamins. So vitamins A, D, E, and K, among others, can only be absorbed while they are dissolved in fat. That also makes these kind of dangerous vitamins because they're actually easier to overdose on. So if these A, D, E, and K, down here it says, will these vitamins be primarily polar or non-polar? So to answer that, we'll see if you can answer it on your own first. But to answer that, we have to look back here. You see that lipids are non-polar organic compounds. So we know polar compounds dissolve in polar solvents, so it makes sense that non-polar vitamins would be soluble in non-polar fats. So they're going to be primarily non-polar. When we talk about these, we have to talk about monomers and polymers. Fats are unique because they have two monomers, and these monomers are not just, it's not going to be one carbon or one hydroxide. This is glycerol. So we see if we write that C Well, I don't need to write it. It's right here, right? So we have a carbon, two hydrogens, one hydrogen on the middle one, and a hydroxide bound to each carbon. So that makes up our glycerol. That right now is completely separated from our other monomer, these fatty acids, which we saw on the last slide. So we have our saturated, monosaturated, and polyunsaturated. So our fatty acids can vary depending on the bonds. When they join together, we are going to, when we draw this reaction, it's moving this way, and we see this 3H2O. Is this water going out or coming in? Are we adding these water molecules, or are they coming out? Another way of rephrasing that, is this a hydrolysis or dehydration reaction? We see we have H1, H2, and O. So we get our H1, H2, and O. So we're removing this hydroxide plus this hydrogen to allow this carbon to bond straight with that oxygen, which we see over here. This is that same bond. So it's a dehydration reaction of removing water and forming this triglyceride. It's always in 3 because our glycerol is always 3 carbons long, and then we're going to add 3 fatty acids to it. Now we said this is dehydration. Is this going to be anabolic or catabolic? Anabolism or catabolism? The way I remember it is bodybuilders take anabolic steroids. They're trying to bulk up. They're trying to get big. They're trying to create more. So when we build up, it's anabolism or anabolic. When we break down, so dehydration, it's also called dehydration synthesis. Dehydration goes with anabolism. Hydrolysis goes with catabolism. Our second type of lipid, phospholipid. It's a complex lipid. It has a phosphate group right here, as well as this head up here, and the lipid section down here. We have a glycerol. Down here we only have 2 fatty acids. Maybe I made a mistake earlier. Triglycerides are always in groups of 3. Phospholipids, we're going to have 2 fatty acids coming off the end of it. So up here, this is water soluble, meaning you should know it's polar. And down here, the fat portion is non-water soluble. It's fat soluble. So this is non-polar. I'm just going to write P. Where have you heard of phospholipids before? Hopefully in Nettie's class. Maybe a little bit in human body systems. I think I mentioned them. We have our phospholipid bilayer on the outside of the cell membrane. Used for repair and construction. It's a component of nervous tissue. This is what we allow those ions, prevent ion movement outside. So remember, we have our sodium-potassium pump. This is where the sodium-potassium pump would be, moving those ions, those cations, from one side to the other. You can see it has these channels through it. I don't need to say anything else. I think you've heard of all these before. Steroids, our third lipid. So they're composed of a hydrocarbon ring structure, which we see when we talk about 1, 2, 3, 4, 5, 6 carbons there. On this one, 1, 2, 3, 4, 5. So it can vary. Most steroids are hormones, not all of them. Steroids are important for regulation of body processes such as metabolism, immune response, reproduction, stress reduction. Cholesterol is the most important steroid. It is the most important because it's used as the building block for all these other ones. We can see what we can see in cortisol, estrogen, testosterone, all of them, they're going to have the same basic structure. We just make changes to what's attached to them. Here we just chop off this whole part, right? Stick hydroxide on the end, good to go. We get a double bonded hydrogen. There's a few changes, but the key is we keep this basic structure, these rings, and change them to create all the different steroids that our body uses. Finally, this is 3 out of 4 proteins. So they're in charge of regulating all bodily function. It makes up 10 to 30% of our mass. Our monomers are our amino acids, and they're connected by peptide bonds. I don't want to get too much into the chemistry of this. Unique properties of proteins determined by the type of amino acids and the sequence of amino acids. So when our ribosomes crank out these amino acid sequences turn into proteins, the type of amino acid that we have, as well as the sequence, is going to change what that protein folds into. And most of what this folding is going on is directly a result of, what kind of bond is that right there? A little hydrogen bond. So everything in red right here, this hydrogen attracted this negative oxygen, this hydrogen. Remember, this is our cation, and our oxygen is going to be the anion. It's these... How did the cat get back in here? Hold on 1 second. I'm going to pause it this time. I'm going to try to pause it this time. No, maybe I'm not going to pause it this time. Yeah, I hear you. All right. Lost my spot. So hydrogen bonds. That's what's managing these shapes of these proteins. We have two categories of proteins. It's going to be structural and functional. Structural proteins, we can see they're going to be generally... I lost my pen. They're going to be long. They're going to be generally nonpolar. They're there to provide, like as the name implies, they're there to provide structure. It's often long repeating sequences. Examples include, should be collagen, keratin, and tubulin. Tubulin's an easy example to think of. When we have mitosis, we get those spindle fibers. Those are made of tubulin. They're helping pull, I guess you could say they're functional in that case, but their structure kind of defines their function. If we go to functional proteins, they are going to be soluble in water. So if structural is insoluble, functional is soluble, which one's going to be polarized? This one. They're chemically active, so they are regulating chemical reactions where these are not. They might be assisting. If you think of tubulin, they're assisting that mitosis, but they're not actually doing anything other than physically assisting, whereas these are chemically assisting. They're often folded into compact specific shapes, so we have a ton of hydrogen bonding going on down here. Some examples include antibodies, most hormones, most enzymes, anything that ends in A's, specifically hemoglobin is another functional protein. So if we look at structural, they're generally used to provide support, shape, mechanical strength to cells, tissues, organs. So collagen is the most abundant protein found in mammals, major component of connective tissue such as tendons, ligaments, skin, and bones. Keratin, fibrous structural protein found in the epithelial cells, including the hair, skin, nails, and the outer layer of the epidermis. And tubulin polymerizes to form microtubules, i.e. the spindle fibers, during mitosis. Structural proteins for the building blocks. Structural proteins for the building blocks of most components of our bodies. That is a terrible sentence. I don't know what I was thinking when I wrote that. But they are the building blocks. You can think of them as providing structure. Functional proteins. Both important, but functional proteins regulate kind of everything we do. So they perform specific biochemical functions necessary for survival. They actively participate in various cellular processes, catalyze biochemical reactions, mediate cellular communication. Some examples. So enzymes decrease activation energy of chemical reactions. Almost, I want to say all, but I'm not a chemistry expert. Most, if not all, the reactions in our body are catalyzed by these enzymes. So what? And it's a specific enzyme for each reaction. There's no enzyme that does two things. It's all going to be one specific thing. So looking at this free energy graph, initially we have our reactants. Then we have to add some sort of energy. Usually this is breaking bonds to change the structure of whatever those components are, those reactants. So we add this energy. We get to this barrier. This is our activation energy, this EA. And once we get to that activation energy, we decrease to form our products. Or we release energy. This is the amount, this delta G is the overall amount of energy that we release. Net energy release. That's probably a better way of saying it, because we're only changing from here to here. We add this energy before it goes away. That's just the activation energy. With a catalyzed reaction, this is the same reaction here, but you see that energy barrier, the activation energy, is much, much less. And so what that's going to do is require less energy for us to complete the reaction. It's going to make the reaction happen faster. It's going to occur more frequently. It's just necessary for just about every bodily function. So decrease that activation energy. We still have the same delta G. We still have the same products and reactants. Just the reaction is much easier to take place. Another example is antibodies. Antibodies respond to foreign antigens such as pathogens or toxins to deactivate or eliminate them. You can think of when we did in, well, y'all didn't have my principles of biomedical science. Hopefully you did some sort of blood typing activities where you looked at the agglutination, which is what's happening here, of red blood cells when these antibodies detect these antigens. I don't know if this picture is specifically supposed to be agglutination or this is just some other foreign invader that our body's taking care of. But that's just my easiest example. Receptors. So here we have a neurotransmitter. We have the synaptic gap right here. This presynaptic cell is releasing the electrical current has come down here, signaled the release of these vesicles with these neurotransmitters. They're coming down here to these receptors that the neurotransmitter is attached to. Those are going to be our proteins down there that are going to bind to specific ligands. So that's going to be an ion or molecule. Specifically here, we're talking about the neurotransmitter to activate a signaling pathway to regulate cellular responses. That receptor gets its appropriate signal and it continues that action potential down the postsynaptic cell. Enzymes, I already kind of talked about them. Function of proteins depends on their structure. So when you're taught this stuff in high school, it's generally pictured as lock and key. So we have these substrates that fit into this enzyme. Active site is the proper shape to receive them. The chemical reaction occurs, these bind together, we have our product. In reality, there's a couple models. We can't observe this, right? It's tiny. So there's a couple models that we kind of better describe what's actually going on. So here we have our enzyme. This active site doesn't actually fit these substrates perfectly, but as it comes down, the active site, it's called an active site because it's going to make changes. It's going to create that shape where it's supposed to be. Those substrate bond to the enzyme. It undergoes another conformational change, and then the substrate or the product is released. The enzyme resets. The key to this is the enzyme is always ends up how it started. So this enzymes don't get used up. Did I write that on here? The enzyme cannot be changed by the reaction and can be reused every time. Protein denaturation. So this is the process by which a protein loses its native structure. It can happen due to extreme changes in temperature, usually heating up, as well as pH changes. So it results in the disruption of those hydrogen bonds. Remember, we have all these hydrogen bonds everywhere that are attracting these coils to each other. Those hydrogen bonds lose their strength for whatever reason, either that temperature or pH change, and we get this denatured protein. The function obviously is lost. You could think of this. Think if this is an enzyme, this shape's completely changing, and it's not working anymore. And we have accumulation of these denatured or misfolding proteins. We call them amyloids. Can cause amyloid diseases such as Alzheimer's, Parkinson's. Recent research suggests that diabetes mellitus type 2 can be linked to certain proteins misfolding. So it's very bad when this happens. And our last organic molecule is going to be our nucleotides. We know it's DNA, RNA. You may have known ribosomes are made out of a combination of nucleotides plus proteins. ATP, the energy source for the body, is a nucleotide composed of nucleotides. Other ones that we'll talk about more later: NAD plus, cyclic. I don't remember what that's called. What does it stand for? It's been a while since I've done any of this biochemistry. FAD, FMN, and a lot of these are part of that citric acid cycle or cellular respiration. Now it's bothering me. I gotta know what CAMP stands for. Cyclic adenosine monophosphate. There we go. So just with the name, you can tell it has to do with that energy production, that metabolism within our bodies. Is that my last slide? I thought I had one more. That's it. I'm done. I'll see you in class.